Hey, hello everybody, and welcome to our module on muscular tissue. In today's class, we're going to talk about the three types of muscle tissue, but we're going to make a special emphasis in the skeletal muscle. So all this part of the class is going to be related somehow with the skeletal muscle. Many of the physiology of the skeletal muscle can be applicable for the other two types of muscle, the cardiac and smooth muscle. And we're going to talk about the cardiac and the smooth muscles later in this chapter, but in more general way. Later in your other courses, especially when you take AP2, you're going to talk about the cardiac muscle in way more detail, and the same thing in the smooth muscle when you see each organ in each system. And then later to end this class, we're going to talk about a little bit about the embryologic origin of the muscles and the stem cells in the muscles. In the human body, we have three types of muscles: a skeletal muscle, cardiac muscle, and a smooth muscle. The cardiac muscle is unique because we can find it only in the heart, so it is unique for the heart. A skeletal muscle are all the muscles you have in your body that attach to your bones. And of course, the skeletal muscle is the type of muscle that allow us to move from one place to another. And if you can imagine, this is the only type of muscle that is under voluntary control. We can control when we move an arm, when we rotate the head, when we lift the leg. Cardiac and smooth muscle, we cannot control when those muscles are going to contract. A smooth muscle, we can find it mainly in internal organs, but also in the walls of the arteries and veins. Muscles has some differences. They have specific structures, but also the three of the muscles share some properties that they have in common. Those properties are here. There are four, and the first one is called excitability. All the muscles, the cardiac, skeletal, and smooth muscles, are made by cells, and those cells contain cells membrane. So in the three of the muscles, the plasma membranes can change their electrical states. So that means that they are able to generate or spread an action potential. When you hear or read the word electrical impulse or electrical states, that refers to the action potential. We're going to talk about that later in this class. Second is elasticity. So a muscle can return to its original length when relaxed. Extensibility, that means that the muscle, it can stretch and extend. And contractility allows muscle tissue to pull on its attachment point and shorten with force. For example, we have the pectoralis major muscle. It's important to know that the muscle contains one side of the muscle is the origin of the muscle. This part of the muscle don't move, or if it moves, it moves a little bit during contraction. And then we have on the other side the attachment point. Now, when this pectoralis major contracts, the pattern of movement is going to go from the attachment to the origin of the muscle. And that is true for the rest of the muscles that are in your body. All the muscles has an origin and all the muscles has an attachment. When that muscle contracts, the movement overall will be from the attachment towards the origin point. Here in this slide, I have some of the difference among the three muscle types, and this is not a detailed list. There are other things that might be not here, but in general, those are the mains that you have to remember. And one of them is, for example, the skeletal and cardiac muscles create striations. Those are absent in the smooth muscle. That's why this muscle actually is called smooth, because it has not those striations. We're gonna see later, we're gonna learn later that those striations in the cardiac and the skeletal muscles is because the type of attachments in the proteins of those muscles, the proteins that make contraction possible, they are linked together in a way that are going to form striations just in the cardiac and in the skeletal muscle, not in the smooth muscle. Other differences here is the skeletal muscle is the only one that has multiple nucleus. So the reason why this is true, or why the skeletal muscle is the only one that contains that, is because during development or embryologic origin, many of the myoblasts, the cells that are going to make muscle, they fuse each other. So they fuse and they make a one only cell, but one only muscle fiber or muscle cell. It actually contains several cells that has been fused, and that's why this type of muscle is the only one that contains multiple nucleus. Cardiac and smooth, they only have one nucleus per cell. The third one, cardiac muscle fibers are physically and electrically connected to each other. So the entire heart contracts as a one unit. That's why I told you later that the heart works as a pump. We're going to see that those intercellular connections between one cardiac cells and another are very important when the action potential travels through the entire organ. Those connections make possible that the heart contract as a whole unit. And finally, the skeletal is a voluntary control, smooth and cardiac muscle involuntarily control. Now we're going to talk about the anatomy of the skeletal muscle. We're going to go from the gross anatomy until the microscopic anatomy, and then to explain the physiology of the muscle, we need to go to the molecular and very, very tiny level. So that's when this topic start getting complicated, because there are things that we might not be able to see with the naked eye. So we kind of need to use our imagination to understand that. And also when we talk about muscle contractions, you have to have this zoom in and zoom out in order to understand how contractions happen. You have sometimes to move out, zoom out, and have a general picture of what is happening, and that way you can understand how the muscle contraction is possible. Now, the anatomy of the muscles. So the muscles in general, this is a skeletal muscle. All the skeletal muscles are wrapped by a connective tissue layer that is called the epimysium. So this is a sheet of irregular connective tissue that wraps each muscle. Inside of each muscle, we have muscle fascicles. So for example, this is 1, 2, 3, 4, 5, 6, and so on and so forth. Those are muscle fascicles. And those muscle fascicles are separated each other's by other sheet of connective tissue. In this case, this is called perimysium. So we have epimysium that wraps the whole muscle. Perimysium wraps the muscle fascicle. Muscle fascicle contains muscle fibers. So this is 1, 2, 3, 4, 5 muscle fibers. And each muscle fibers is wrapped by endomysium. So this is an important thing that you remember, that we have those 3 layers of connective tissue. And why those connective tissue are important? They're important because keep the muscles organized. So from outside to inside, we have epimysium the whole muscle, perimysium, the muscle fascicle, endomysium, the muscle fiber. Now in this level here, the next picture, it will show you the myofibrin. This is the place where all these myofibrins that form the muscle fiber, this is the place where contraction will take place. Muscle contraction will take place. Now, when you see the word muscle fiber, I want you to think about muscle cell, because a muscle fiber is equal to one muscle cell. So a muscle fiber, one muscle cell. You can see here the nucleus. And those muscle fibers or muscle cells, they can be very, very long. So they can be short, they can be long. That's going to depend on the type of muscle that you have. So let's pretend that this is a biceps brachii in the anterior part of your arm. So you know that how long is your biceps just by looking at it. So that length is the length that has each cell. So if in your case, your biceps has like 30 centimeters or I don't know how many inches, it don't matter. How long is your muscle? That is the length of each individual muscle fiber. And then if you look closely this picture here, don't confuse these myofibrils like the only cell. Those are not a cells, those are part of one cell. And importantly is that the cell membrane in the muscle cell is called sarcolemma. This is the equal to the normal cell membrane. So sarcolemma in a muscle cell, cell membrane, and the rest of the cells. And inside of that cell membrane, that sarcolemma, we have organelles. We have the nucleus, we have sarcoplasmic reticulum, we have mitochondrion. This here, that those are important for energy generation. And then we have the myofibrins there that are the ones that are important for muscle contractions. I mentioned this early, that muscle cells are long and cylindrical. They can be long up to 30 centimeters, and your greater muscle, like in your sartorius muscle and the quadriceps femoral muscles, so they can be pretty long cells. Just one muscle fiber can be that long. So just keep that in mind so you have a picture of how long is a cell. And this is the representation. So of course, this is not a scale. But remember that this is a whole cell that contains multiple nucleus, and the membrane, the cell membrane, is called the sarcolemma. And then we have mitochondria inside, we have other organelles. This space here is the sarcoplasm. Sarcoplasma is the equivalent to the cytoplasma in a regular cell. And inside of the cell, we have the myofibrins. Those are important because the myofibrins, we can find those in the cardiac and the skeletal muscle, but in the smooth muscle, because the proteins that contracts, they are organized in a different way, we cannot find myofibrils in the smooth muscle, just in heart and the skeletal muscle. I told you this early, that the reason why the skeletal muscle has multiple nucleus is because the myoblasts fuse during development. All right, now we're going to talk about the myofibrins in more detail, and that's when the things start getting complicated, because we're going to the point that is very, very small. So even even smaller than a cell. So when we take one myofibrin and then we zoom in, we can see this. Now, it's important that you remember that the skeletal and the cardiac muscle has those striations. that you can see it here, like those dark rings around the cells. Those dark rings are called A band. Take a look closely. Here in the myofibrin, if you look closely, the sarcomere, which is this part here, are the ones that are responsible to form that A band, which again is the one that is going to form those dark rings. They're the striations. So you can see here that the sarcomere is the functional unit of the muscle cell. Why is the functional unit? Because contraction is going to happen right here. So sarcomere is the functional unit of the muscle because contraction will take place inside of the sarcomere. The sarcomere, you can see here, we have a C disc and a C disc. It's called a C disc because it has this zigzag pattern shape. This Z disc is made by proteins, and these proteins, the important ones you need to remember is this one here that is green and thin. That's the actin. And so the sarcomere is the space between two C discs. Right in the middle, we have not just actin, but also we have another type of protein in the middle, and that is called myosin. So because we have so many proteins concentrated in this area, that is what is going to make those rings that looks more darker. And that's what is going to make those striations. If you look the space between sarcomeres, for example, this is another sarcomere here, and we have another part of the sarcomere here. So the spaces between the sarcomeres are the lighter part of the muscle. All right, and let me clean this a little bit. All right, now that we have this clean, let's recap really quick. So the sarcomere is the functional unit of the muscle fiber, and inside of the sarcomere we said that we have the C disc. In the middle we have the M disc. M because middle. So contraction, essentially, you think about when a muscle contracts, what is happening is that there is a short. If you think about the whole muscle, if I just pretend that this is a muscle, this muscle is relaxed. When this muscle will contract, of course, this muscle will look more like this, has been contract. So that contractions at the cellular level, at the almost molecular level, will take place right here. So if you look here, the purple ones, I told you those are myosin, and the green ones are actin. Those two filaments will connect or attach each other, and then myosin will pull the actin filament towards the middle, towards the midline. When that happens is when contraction occurs. So when this muscle fiber contracts, essentially the distance between C discs, it will be short when the muscle contracts because the myosin is pulling those actin filaments towards the middle. And that, if you can imagine that that's happening to every sarcomere, the final result is that the muscle fiber is contracting, getting shorter, and if that happens, in all the fibers, the muscle fiber will be short, and if the whole muscle fiber is short, the muscle fascicle will be short, and if the muscle fascicle will be short, the whole muscle will be short. That is contraction. I mentioned this early. Sarcolemma is the plasma membrane in the muscle fiber, and sarcoplasma is the cytoplasma of the muscle fiber. Other crucial and very important organelle that you have to know in order to understand the physiology of muscle contraction is the sarcoplasmic reticulum. This sarcoplasmic reticulum is a specialized, smooth endoplasmic reticulum. In this picture, we can see that is in yellow, represented in yellow. And don't think that this sarcoplasmic reticulum is that small. Really, if you put close attention here in the top, this sarcoplasmic reticulum has been cut. So you can see all this part, but the reality is that this sarcoplasmic reticulum extends all over. So if let's move back here, and if I want to draw the sarcoplasmic reticulum, it will be wrapping the whole thing. So it is around the whole myofibril. And the sarcoplasmic reticulum, we're going to talk about that later, but and you will notice that in this class I'm going to repeat things. Hopefully that will be good, so you can remember things better if I repeat it. So just for now, just remember, sarcoplasmic reticulum is a very specialized, smooth endoplasmic reticulum, and the main function when we talk about this in contraction is that this organelle contains calcium. Is the main place where we have calcium storage, and that's going to be very, very important for contractions. Other part, the sarcomere, I told you this, that contains the actin and myosin, and that is what make those striations in the skeletal and cardiac muscle. And the sarcomere I mentioned is the functional unit of the skeletal muscle, because this is the place where those two proteins are going to attach each other, and one is going to pull the filament, and that will make contraction. That's why it's the functional unit, because contractions happens right here in the sarcomere. Now let's talk about the functional unit of the muscle fiber in more detail, the sarcomere. So the sarcomere, each of the sarcomere is bordered by structures called C discs or lines, which are those two here. I told you this is a closer lookup of this. The sarcomeres are the ones that make this A band that makes the striations. Those C lines contains that green thin filament that is called actin, and we're gonna see that actin and contains also other type of proteins that are kind of related. Those are troponin and tropomyosin. So those troponin and tropomyosin form a complex because they are attached each other. We're gonna see why those proteins are important for contraction, for muscle contraction. The actin filaments also, if you remember those C lines that we can see here, when I'm drawing here in the right side of your screen, from there you can see those projections that the C lines, those are the actin filaments. And if you put attention, the actin filaments, they don't get too close to the midline. So that is called the H line. And the distance between one actin and the other actin is called the H line. You don't have to really remember this. Just think about the midline, the M line, which is right in the middle of the sarcomere. Now there in the sarcomere, we have other type of filament, those are thick filament. This is myosin. Now the interaction between thin and thick filament is what make contraction possible. Myosin pulls the actin towards the midline, and that translates to contraction. So you can see here that this in purple is the myosin, and the myosin contains those heads here. This is the head of the myosin. This portion here is important because it's flexible, and this is actually what is going to make or will pull the filaments of actin once this head attached to the actin. If you can see here, this is a closer lookup of the actin protein. We can see the troponin here, which is this, and we can see here better. So the light green is the actin proteins, and green dark, those are the binding sites for myosin. This is the place where the head of the myosin will attach to the actin. And this head, remember, is flexible. This is the part that is going to move or pull the actin molecules. Now, between those, we have those proteins that we talk about, the troponin here and the tropomyosin here. So in a muscle that is relaxed or in a relaxed state, this tropomyosin and troponin will be blocking the binding sites of myosins. Therefore, contraction will not take place. But eventually, and we're going to see later, there are going to be a sequence of events that are going to make possible that those troponin and tropomyosin move away from the binding site, and myosin will be able to attach to actin, and contraction will happen. All right, now that we talk about the sarcomere, let's zoom out just a little bit. Remember that we just talk about the myofibril in the muscle fibers. Remember that in this part is the place where we can find those actin and myosin filaments. So now let's zoom out and look the place where the muscle fiber, remember this is a cell, there's a muscle cell. So where's the place where this muscle cell actually make contact with a neuron? This is the axon of the neuron. And if we keep drawing this, we're going to get in some point where we can get the body of the neuron with the nucleus, and we have those dendrites. So anyway, this is a neuron. Why this is important? Because the only way that we can make a muscle contract is towards the neuron. If a neuron do not stimulate a muscle fiber, the muscle will not contract. You need a neuron in order to contract a muscle. So that's why this part here is important, because that's the place where the neuron connects to the muscle fibers. These connections are in all your body where you have a skeletal muscle. There should be a neuron that it will innervate the muscle. Now, this neuromuscular junction is the site where the motor neuron's terminal meets to the muscle fiber, that specific part here. And cells has membrane potential or electrical gradients across the membrane. We're going to see that both neurons and muscle cells can generate action potential through the membranes. And as a general, keep this in mind, all the cells, especially in this case muscle cells, normally inside of the membrane, inside of the cell membrane, remember cell membrane in the muscle cell is called sarcolemma, and we have there an action potential, we have there a negative charge inside of the cell. So if I draw here the sarcolemma, here we have the sarcoplasma, which is the cytoplasm. This inside of the cell, this is the intracellular space, and this will be outside of the cell, so this will be the extracellular space. Inside of the cells in general has a negative charge, So overall, this negativity is something around minus 60 or minus 90. And as in general, the extracellular space has a little bit of positivity. So when we change between negative and positive, that is called an action potential. Here in this slide, I'm going to explain a little bit in more detail the action potential. Why this is important, because if you, more than memorizing, if you just understand the action potential, it's going to be easy for you to understand the process of contraction, because contraction is a pathway from the neuron to the muscle to the sarcoplasmic reticulum to the filaments that contracts the muscle. So first of all, both neurons and skeletal muscles, they can generate action potential because those tissues can be electrically excitable. Other thing is the neurons and muscles can use their membrane potential to generate electricity or electrical signals. Those are the ones that we call action potential. And how the way to do that is when they control the movement of particles that have a charge. If you remember from previous classes, an element that contains an electrical charge is called ion. So if you can imagine, if you can manipulate in some way how much of those ions, either positive or negative charge, come in or come out through the cell membrane, that will generate an action potential. All right, so to explain this, I'm going to draw here this. Let's pretend that this is a muscle fiber. Remember that the muscle fiber contains this sarcolemma, which is the muscle membrane. So I'm gonna make it more circular, more like a regular cell, to make it just clear. So we have a nucleus, we have multiple nucleus here. I'm just gonna use one nucleus. We have the cell membrane that wraps or is around the cell. Right now, I'm gonna take this little part and that little circle is gonna be this. Now notice that I'm leaving some gaps. This is because I'm going to draw something later. So that little portion, this is inside and this is outside, or intracellular space and extracellular space. So here we have the intracellular space and we have here the extracellular space. Now I left those gaps because I'm going to draw here some structures that are going to be important. Those are proteins. Remember when we saw the class of cell in the membrane, we have integral proteins. Some of the proteins work as a channel, so it will allow things, substance, cross the cell membrane from one side to another side. And remember, in some point, I told you that those ions, when we talk about ions, sodium, potassium, calcium, those ions have concentrations, and inside of the cell it has different concentrations outside of the cell. So when we talk about also diffusion, we talk about that in general things move from the greater concentration to a lower concentration. So if you have more sodium in one side, by simple diffusion it will move from the higher concentration to a lower concentration. So as in general, we have more sodium outside than inside of the cell. I don't know if you remember this, but I'll tell you, we have a special pump when we talk about active transport. Active because we use ATP in order to make this. There's a pump that is moving sodium from a lower concentration to a higher concentration, and to do that, because it's against the concentration gradient, you need energy, ATP. So this sodium potassium pump, it will move sodium from a lower concentration to a higher concentration, and that's why sodium most of the time is in greater concentration outside than inside, because we have this pump that is pumping sodium from inside of the cell to outside. That's why we have more sodium outside than inside. All right, anyway, here what happened is I told you that inside of the cell we have this negative charge or voltage. I'm gonna make this as a negative here. We said that it's around -60 or -90. In general, it's around 70, I think. It don't matter too much, that number. You know, the thing is, if it's more negative, you will need more positive charge to make this number more positive. If it's closer to 0, like -10, that means that you just need 10 positive charge to make it 0, right? That's simple there. All right, so in general, we have this that is more negative. We have more proteins. Proteins in general have negative charge. So that's why inside of the cell also is more negative. And outside of the cell, it tends to be more positive, because you remember what is inside? All of this potassium, which has a positive charge. All right, now, what happens when you have an action potential? Something triggers a change in this homeostasis. So something like, for example, imagine, let's take as an example this, because we're going to see this later, how that works. This pump, let's pretend this is a sodium pump. So this sodium is a channel. I'm sorry, I say pump, but let's pretend this is a channel. And this channel right now is closed. The only way that you can open this channel is as a hormone or a substance or a ligand attached to this receptor or to this channel. So this substance attached to this channel. When it's attached, this channel opens. Once this channel opens, sodium, because it's in greater concentration outside, it will flow from outside to inside of the cell. So sodium, because it has a positive charge, is going to start raising this negativity here. So this will become a little bit more positive, a little bit more positive, a little bit more positive, because positive charges are getting inside from the sodium. Now here, positive charges in this side, the extracellular space, positive charge are leaving the extracellular space. So this part is becoming a little bit more negative, 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 and this more positive, positive, positive. So see how this start changing. So this positive is going to start moving in this direction, and this negative in this direction. So we see that we have negative here, and we start seeing positive here. Now this other is another channel, it's another sodium channel, but this channel is different. This channel, you don't need a ligand like this in order to activate. This channel is a voltage channel. So in other words, when there's a change in the voltage, like the one that we're seeing here, that changes between negative and positive to negative, this channel is going to be active when that happens. It's a voltage gauge channel. When that happens, because it's a channel, it will open, and therefore we have more of those channels. So it will open, and that will allow the more sodium go inside of the cell. And that's going to make overall that, now I want you to look back here, if the action potential is started in this point, it's going to start spreading all the way to the cell membrane. Right? And that is the action potential. When this moves in this direction, the part that is getting behind is returning to the normal point where you have positive charge here, and here you have negative. When this action potential is moving, this is becoming positive when the action potential is passing through there, and then this is more negative. So think about the action potential like a wave in the ocean. So we have negative here, the action potential is moving in that direction. Think about the action potential like a wave. And then after that, we're going to see that how this action potential actually is very, very important for muscle contraction. All right, in this picture, I have the general pathway that you have to kind of remember. We're going to go in detail, and we're going to divide this in two parts. This is going to be part 1. This is going to be part 2. But the thing is, the events that happens when a cell, and specifically when the neurons, is activated and is sending electrical impulse. Remember, electrical impulse, think about action potential. So first, the neurons will send that nerve impulse or action potential, and it's going to go from the body of the cell. It's going to travel through the axon of the neuron until the end, the dendrites at the end. And you remember that we have the muscle here, and this connection between the neurons and the muscle is the neuromuscular junction. So the nerve impulse will go through this part, to the neuromuscular junction. Then the action potential, we're going to talk about in more detail, is going to move to the muscle fiber. In the muscle fiber, the action potential will spread until reach the sarcoplasmic reticulum. Once the action potential reach the sarcoplasmic reticulum, that will activate the sarcoplasmic reticulum, and that will cause the calcium inside of the sarcoplasmic reticulum is released. Once the calcium is released, this is going to go, or this calcium is going to be released in the cytoplasm, or sarcoplasma in this case, because it's a muscle. When we have all this calcium will join or will attach to the troponin and will form calcium troponin complex. When the troponin got the calcium, it will change in shape, and the troponin that initially was blocking the binding sites is going to move. So it's going to leave those binding sites free. When that happens, the acting binding sites attach to the myosin heads. When the myosin heads are attached, those heads will pull the actin filaments towards the center, towards the M line, and will cause muscle contraction. So that's the big picture. And the next slides, we're going to talk about this part first, and then we're going to talk about part number 2. Now let's talk about the excitation contraction coupling. This essentially is going to be the place how the action potential travels from the neuron to the muscle fiber. I told you that neurons are actually very long cells. So for instance, if you think about how big is a neuron, how long is a neuron, just think about if you want to move your big toe, you only need 2 neurons in order to order the muscle to move. So you have one neuron in your head, that neuron in your head, the body, is in your brain, and then the axon, it'll prolong, so it will extend all over your brain, go down over your spinal cord, and then in your lumbar spinal cord, this first neuron will make contact with the second neuron. So let's pretend this is neuron number 2, and neuron number 1, it just make contact here. Let's pretend that the body is here, and you have your brain here. You have your spinal cord here. So this first neuron will make contact with the second neuron in your spinal cord. And then from there, the body of that neuron, the second neuron, is in the spinal cord. In the axon, it will extend all the way to your leg until the muscles in your big toe. So just imagine how long it is. And then finally, the last part of that neuron will touch the muscle fiber that needs to be moved. Muscle fibers, again, also are very long, depend on the muscle. If you have muscle that is big, the biggest is the cell. In your big toe, if you have muscle that is going to move, it's not going to be that long like the quadricep femoral or the biceps brachii, but it's going to be long. Anyway, so here, now we're going to focus here what happened in the neuromuscular junction. So we said that the action potential travels to the axon all the way to that neuromuscular junction, and then there the axon terminal will release acetylcholine. Now if you look here, we're going to take a closer look here, zoom in. The action potential is travel, remember the action potential is travel all the way to the membrane, and this action potential, it will cause in the neurons that some channels, especially calcium channels, opens. Calcium will get inside of the neurons, and that will cause that all these vesicles release the content outside. So remember, the action potential, go through there, activate those calcium channels. Calcium channels will go from outside to inside. That will make these vesicles release by exocytosis release the content. And what is the content? If we look closely, that content is acetylcholine. Now this is a neuron here, and then we have a muscle fiber here, which is the muscle cell. And I want you to look two things. First, this substance is called a neurotransmitter because it's released by the neuron, and that neurotransmitter, it will attach to the receptor in the muscle fiber. So that is specific for that. This acetylcholine receptor, we have it, let's see, I don't have it, yeah, so here, this acetylcholine receptor is located in the sarcolemma, which is the cell membrane, and will cause despolarization, or it will cause changing in the charge, in the electrical charge in the muscle. Because if you can see here, this receptor is actually a sodium channel that it will open only when the acetylcholine attached to this receptor. Once this complex acetylcholine receptor forms, the channel will open, and remember, sodium is in greater concentration outside than inside. So sodium will flow from outside to inside. For sodium has a positive charge, so the negativity that is inside of the cell will change to positive, and that is action potential. And then that action potential, it will spread through the whole cell membrane. And I told you this when I was explaining you about the action potential. There are some channels, there are voltage gate channels. Those channels will activate when the action potential reaches them. And in this case, it will open more sodium channels, more sodium, more positive charge are going to go inside of the cell, and that's just going to trigger that the action potential is spread through the whole sarcolemma and the muscle fiber. Now, other important thing you need to remember is that we have this acetylcholine. We need something to regulate that. If you have acetylcholine that keeps stimulating those receptors, you will have contraction in your body, like without control. I mean, if you have acetylcholine, it's just going to keep activating your muscle. So you have to have something to contrast that acetylcholine, and that is called acetylcholinesterase. So that is enzyme that will remove acetylcholine from the neuromuscular junction, and it will prevent muscular contraction. So when we want contraction, we're going to release acetylcholine. When the contraction is no longer needed, we're going to have acetylcholinesterase that will remove that acetylcholine, and then those receptors will be closed again. All right. Now I'm going to make a sketch about this muscle fiber. So remember, muscle fiber is the whole thing here. So what we are looking here in this picture is just a tiny fraction of the cell membrane that, if you remember, is called sarcolemma. And then inside of the muscle fiber, we have all those myofibrils. They're also like a tubes, which all those myofibrils are those little tubes that we can see here. So wrapping those tubes, we have the sarcoplasma reticulum. Remember, sarcoplasma reticulum, I told you, is important because contain calcium, and calcium is crucial for contraction. So far, we know, just pretend that this part is the part that we are looking here. And let's just pretend that the neuron, the neuromuscular junction, is here. So what happened is acetylcholine was released here, sodium channels opens, and the action potential started here, and it started spreading in this direction, and then here in the sarcolemma, I want you to look those spaces, those foramens. Those are important because those form a structure called the T-tubule. And what this is going to do is that it's going to allow the action potential travel from here, from the surface, going down here, and then spread that action potential to the sarcoplasmic reticulum. So this triad between two sarcoplasmic reticulum and a T-tube, that is called a triad, and that's why it's called T-tube, because of that. But also you can think about the T-tube like this T shape. It has like a T shape. Anyway, those T-tubules are very important because they help to spread the action potential from the cell membrane, sarcolemma, to the sarcoplasmic reticulum through those structure. Once this action potential reach the sarcoplasmic reticulum, it will activate. And I'll tell you, you don't need to know that, but what is going to happen is that it's going to activate some channels. There are voltage channels, but instead of being sodium, they're calcium voltage gauge channels. So those channels will be open, and if I told you that this is the place where you have more calcium because it's a storage, that means that inside of the sarcoplasmic reticulum you have a lot of calcium. So once you open those channels, calcium will leave that sarcoplasmic reticulum and will go through that cytoplasma. And once the calcium is in the cytoplasma, these myofibrins, we're going to see that the calcium, it will attach to the troponin that is in your actin. All right, let's summarize this. So the action potential started in the neuron, then it travels through the axon to the neuromuscular junction. There in the neuromuscular junction, the action potential makes possible the release of acetylcholine, which is the neurotransmitter. Acetylcholine will attach to the receptor in the muscle fiber, so that's the acetylcholine receptor, that in the same time is a sodium channel. So when the acetylcholine attached to the receptor, it will open. Sodium will be able to get inside of the muscle cell, and that will make possible the action potential spread from the nerve through the muscle. once the action potential reach the muscle, that will spread all over to the muscle fiber until the T tubules. In the T tubules, remember that we have the sarcoplasmic reticulum in the sides. So the sarcoplasmic reticulum will be activated because they have some calcium channels. There are voltage gate calcium channels. So when the action potential reach those channels will open, and calcium, which is in greater concentration inside of the sarcoplasmic reticulum, will move to outside. So calcium will be released from the sarcoplasmic reticulum to the cytoplasma, in this case, cytoplasma, sarcoplasma. And once that happens, we're going to see this part next. The calcium will join to troponin complex, to troponin, and will form the calcium troponin complex. All right, to understand the next phase, at this point, the muscle has not been contracted yet. Now, I want you to think about all this process of contraction. All this process happens in fractions of seconds. So even though it's taking me almost an hour to explain this, in real life, this happens. Just think about how long it takes from, when you think about moving your arm, and when you actually move the arm, it's less than a second. So all these things are happening very, very quickly. So I want you to, think about that. Of course, in order to understand this, we're kind of going in slow motion, very, very slow motion. All right, so we talk about the calcium and why the calcium is important. To understand also this, I want you to put attention in this picture. Remember, this is a myofibrin. So that myofibrin we have many myofibrins that are part of the muscular fiber, and the muscular fiber is the cell. So those myofibrins contains those proteins, actin and myosin. Actin is this in purple. Excuse me. Myosin is this in purple, and actin is this in green. Remember this line, the midline, is very important. And remember that the distance between two C lines, that's the sarcomere. That's this part here, that is the functional part, because this is the part that contracts. So when contractions happens, the heads of those myosins attach again to the actin, and then will pull the actin in this direction. It will pull towards the midline. We can see that representation here. See the distance here, and then see how they look when it's contracted. So that contraction, the C lines, they're getting closer each other when the muscle is contracting. Now this has happened in a single sarcomere, but if you can imagine, all the rest of the sarcomeres are doing the same thing. So overall, what happened is that the whole muscle fiber overall will contract. So it's a chain of sarcomeres that has been contracting, that at the end will make the whole thing, the muscle fiber contracts, and then eventually the whole muscle will contract as well. And that is called the sliding filament module, and it's called sliding because you can see that the actin slide towards the midline because the myosin is pulling the actin towards the midline. So that's why it's called a sliding filament module. I told you that this binding or union between actin and myosin only happens when the binding sites of the actins are exposed. I told you that we have tropomyosin and troponin that form that complex, and that complex is the one that prevents the myosin attached to the actin. Now, the importance of the calcium, if we keep following the path, the importance of the calcium is that the calcium is the one that is going to allow is going to attach to the troponin and it will make the tropomyosin and troponin complex that has been preventing myosin attach. It will change the shape of those proteins and the binding sites will be exposed once calcium attach to troponin. Now, other thing that is very important that you remember is that in order to have muscle contraction, we need calcium, and of course, we need ATP. ATP, adenosine triphosphate, is energy. So this is a molecule that the body uses in order to have energy. So I told you that the heads of the myosin will pull those actin. You can imagine that's a work that requires energy, and energy will be the source of energy that myosin will use is storage and the bonds here in the ATP molecule. Okay, in the next slides, I'm going to show you how that head of the myosin attached to the actin, how that after that pulls the actin towards the midline and contractions happens. So to do that, first I want you to look this picture. I want you to look here, the M line, and the part that we are seeing is this one. So this is this, and this myosin that we have here is this one. So when this head pulls, the midline is going to be here. I just want you to keep that in mind, that midline is going to be in this side. So when we don't have calcium, when the muscle is relaxed, all those binding sites in the actin has been blocking by the troponin and the tropomyosin. So even though the head of the myosin wants to get there, it's not possible because it's blockaded. It's blockaded. Once you have calcium, remember, that's what happens when you have calcium. All those white parts are calcium. The calcium will attach to this troponin, and that will change the protein shape. So it's going to twist a little bit more once calcium touches troponin, and that change will make available those binding sites. So troponin, calciotroponin will, calcium troponin complex will then make that the tropomyosin moves away from the binding sites. Once that happens, the myosin head is almost ready to attach. Now, all this is happening like very fast, like I said, and it's not, it's not that it's happening first days and then days. For example, here, in order to have this head ready to attach, this head of the myosin, first it has to use ATP in order to get this kind of a stage in which there's a lot of energy trapped there that is going to be released once this head attached to the actin. But you can see here in yellow that has ADP and phosphate inorganic. That's what the P is for phosphate, the I is for inorganic. Remember, ATP means that we have 3 phosphate. If we remove a phosphate from the ATP, we have ADP. And that's what we have here, that ADP, and that phosphate that we remove is the phosphate that is here. So every time that we break that bond, energy was released. So that energy was the one that the head used in order to make this part of the head, like, just think about the rubber band. So we use the energy when we stretch the rubber band, and when we, in some point when we want to release the energy, the rubber band will back to the point, to the original point and more. So think about this like a rubber band. So we use a little bit of energy in order to stretch this head, and then following that is ready to attach to the actin. You can see here that this is the following. So we stay here In this point, this is called when the myosin head is in this high energy level, is called the cocking of the myosin head. After that is when the head and when the binding sites are available, this head moves from there to here, and then when it's attached at the end, the flexible part, it will move this myosin, this actin towards the midline. Remember, the midline was in this side according to the picture. So it will pull this actin towards the midline. That will cause contraction or shortening of the C discs. That movement here is called a power stroke. And of course, it requires that energy that was trapped, energy that was storage still. And when that power stroke is released, those ADP and phosphate are they're released as well. Now, other thing that is important is that cross bridge that's the name they receive when the myosin and actin are together. It's called cross bridge. Now, when that power stroke happened, then one part of the myosin head, remember that is the part that is attached to that binding side of actin, but the head has another binding side that is made by ATP. Now, ATP binding causes the myosin head to detach from the actin. So if you look here at the picture, after the stroke, this union between the head of the the head of the myosin and actin is very strong. So it's not easy to separate them. So the only thing that it can separate is energy. So therefore you need another ATP molecule in order to generate the energy that will break that union. So once you have that, you break that, and that energy will be used to form this cocking myosin again. So the energy that you use to separate is good enough to make the head of the myosin ready to complete the cycle again. Other thing is that as long as ATP is available, this attachment and cross bridges cycle will continue occurring, and muscle contraction can occur. In absence of ATP, the myosin head will not detach from the actin. This is important because when people die, for example, the heads of the myosin, they cannot be detached, because when you die, you don't have more ATP. All your organs, all your mitochondria, your lungs that used to get oxygen to form ATP, when you die, you lost your ATP. And remember, the ATP is requirement to break this attachment. So when somebody dies, for example, you don't have ATP and your muscles start getting stiffed. So that's why people who die after a few hours, the arms and the muscle get stiff and contracted because of this. You need ATP in order to relax your muscles, but also you need ATP in order to contract. So if we think about a cycle, you need overall 2 molecules of ATP. One, the one that you're going to use to form the cocking of the myosin head, which is this one, and then the other one, the other molecule that you're going to use to release the head of the myosin from the actin. Finally, this is an overview of contraction and relaxation. Remember, contraction, you think about moving a muscle, a neuron will go from your brain to the spinal cord, other neuron will move from your spinal cord to the muscle you want to contract. The action potential will travel from your brain all the way down to the neuromuscular junction. In that neuromuscular junction, acetylcholine will be released, and those sodium channels will be open, and that way the action potential will spread from the neuron through the muscle fiber. Then the action potential will keep going until it reaches the sarcoplasmic reticulum because of the T tubules. Once the action potential reach that sarcoplasmic reticulum, sodium channels that are sensitive to those voltage, it will open. Sodium that is in more concentration here inside will be released outside by simple diffusion. All those calcium that is being released in the sarcoplasm or cytoplasma, if this will be a normal cell, it will attach to the troponin. Once the calcium attached to the troponin, tropomyosin moves away from the binding sites. Once that happens, also we has been already used an ATP in order to make this head of the myosin a high energy form. Once those binding sites are free, the head attached because there's a very strong attraction, and then the ATP is going to release the energy. So this head will move towards the midline and will cause muscle contraction. When the opposite happens when we need to relax the muscle. If you think about relaxation, like if you think about moving your muscle to do something, and when it's done, you don't want your muscle keep contracting. So what is going to happen is the signal is going to go away, so no more acetylcholine will form. Then we have a sodium pump here that will use energy to move the calcium from, I say sodium initially, is calcium. So we have those calcium pumps that are active transport because they're going to move calcium from this concentration, and they're going to start moving from a lower concentration to a higher concentration. So to do that, you need energy. Anyway, so in relaxation, all the calcium is moving away from the acting, and therefore the troponin and the tropomyosin will block again the actin binding sites, and relaxation will occur. Again, those process of contraction and relaxation, all this process happens in fractions and fractions of seconds. Just it's like when you think about a movement, a muscle, everything happens so fast that there's not almost a lag between you thinking moving a muscle and you actually moving a muscle, and then thinking about relaxing the muscle and when you actually relax the muscle. So all this happens very, very quickly. I told you earlier that we need two substances that are crucial for muscle contraction. One was calcium, and of course ATP is the next one. ATP because that's the energy that we use in order to perform things. To make things move, we need energy, and that energy is stored in the bonds between the adenine and the phosphate. So there's some mechanisms that the body uses to get or regenerate ATP. The first one is called the creatine phosphatase metabolism, and then the second one is called glycolysis. So in the first one, the creatine phosphate metabolism, creatine phosphate derivate ATP powers the first few seconds of muscle contraction. So this is how the muscles use or generates ATP for the first 15 seconds. So if you are thinking about moving a table, the first 15 seconds, the ATP that you're going to use is going to be coming from this creatine phosphate metabolism. And what is that? You can see it here. We need a substance that is called creatine, and we have this creatine that is going to attach to the ADP molecule. Now, how this happen? We need to attach. We have ATP 3 phosphates. when we need to make more ATP, normally what we have is ADP. So the goal here is to add another phosphate group. So this ATP, let's see that you use ATP for something, you break the bond and one phosphate will go this way and the ADP will go this way. So we have the ADP, we have the phosphate. That phosphate that it just has been released from the ATP, it will join the creatinine and will form this substance, creatinine phosphate. And it's called creatine phosphate because it contains a phosphate group. Now here's the part that is important. We have an enzyme in our muscles that is called creatine kinase. Remember that ACE is an enzyme. And that creatine enzyme will remove the phosphate from the creatine phosphate and will attach that to the ADP. So that way you form again a molecule of ATP and creatinine. Now, like I said, this method is very efficient, but it just lasts 15 seconds. And if you think about creatinine, there's a lot of people who lifting weight or do a lot of exercise. Sometimes they use supplements. You're gonna see there are supplements that contains creatinine, and that's why they drink those things, because if you, in theory, if you have more creatinine, you will have more endurance and will be able to contract without fatiguing your muscles for a longer period of time. Now, in the second method, the glycolysis, remember that glycolysis is when we break down glucose. So if we have here this picture, we can get glucose through the blood when we eat, or glucose that has been stored in your liver in form of glycogen. So anyway, when the muscle is contracting, let's say you're doing exercise, you need glucose in order to get energy. So that glucose, if you remember, is the simple sugar. When we break down the simple sugar, the sugar is no longer a sugar. But in the process of breaking down this glucose, we will generate 2 molecules of ATP and 2 molecules of pyruvate. So when we break 1 molecule of glucose, we got 2 ATP that we can use for muscle contraction. Now here's is going to happen 2 things. One is if we have oxygen available, we're going to use that oxygen to perform something that's called aerobic respiration. We're going to see in a minute that happens in the mitochondria. And from this point, we're going to generate a lot of ATPs through this process. So this is the most efficient way to create or generate new molecules of ATP. However, we need oxygen and we need pyruvate to do that. The other way is that we don't have enough oxygen. What is going to happen is the pyruvate, without presence of oxygen, it will take another metabolic pathway, and we still might be able to generate some ATP, but the bad part is that as a residual product or waste product, we will have lactic acid. Now, lactic acid is might be contribute to the muscle fatigue. So if you have lactic acid building up in your muscle because you're not receiving enough oxygen, let's pretend you're doing exercise, lifting weight, doing a lot of exercise, the oxygen that you're breathing might be not enough to compensate, the necessity for ATP. So one portion of the oxygen you need is going to go through aerobic respiration, but then the other of the molecules of glucose might be through anaerobic respiration. And in that case, anaerobic means not oxygen, you will have lactic acid. So if you do exercise in the gym for a long period of time, your muscles start feeling like burning and fatigue. That's because you're building up lactic acid. Aerobic respiration, we use oxygen, so it's when we break down glucose and other nutrients in presence of oxygen. The glucose from your blood, the pyruvic acid, and the fatty acid, they go to the mitochondria, the products, and then we combine oxygen in a very complex process that we're not going to get into details here. But the thing is that you need oxygen in order to perform aerobic respiration, and at the end, the mitochondria, it will provide, if you can see, a bunch of ATP. It will generate a little bit of heat. Those are wasted products. But the important part here is that this is the most efficient way. Now, of course, if you think about zoom out, when you do exercise, you use those three. You don't use just one, you use a little bit of the three. The first seconds you use this. When you ran out of creatinine, you start using glycolysis, and you use a combination of both processes. You use aerobic respiration because your respiration, you are getting oxygen, but the amount of oxygen that you breathe is not enough to for the demand that you're gonna. Part of that metabolism also is going to be anabolic, and therefore you're still going to form some lactic acid that it will fatigue your muscles when you keep doing exercise for a long period of time. All right, at this point, I will suggest you take a break so you come for the next part with a fresh mind, and we're going to talk about how the nervous system controls your muscle tension, and we're going to talk about some of the type of muscle fibers, exercise and muscle performs, and when we finish that part, then we're going to talk about cardiac and smooth muscle. When we talk about muscle tension, is that all the tension the muscle generates in order to contract. So the force generated by contraction of a muscle is a muscle tension, and we have two types of muscle contractions. Those are the ones that are important: the isotonic contractions and the isometric contractions. Essentially, the isotonic, the muscle produces tension, changing the angles of the skeletal joints. And the isometric contraction, the muscle produces tension without changing the angle of the skeleton joints. So I'm going to explain this. I want you to look at this picture. If you have this humerus, and then we have here the four arm bones, the radius, ulna, and this muscle, when it contracts, before contracting, you have an angle here, right? Let's pretend this is a 90-degree angle. When the muscle contracts, it will pull the bone towards the humerus, and now the angle change. So let's pretend that this angle is not 90, but I don't know, 80. So that the angle has been reduced or changed. That is isotonic contraction. There was a movement associated with the contraction, and sometimes we use load for that. So we move the load from here to there. So that's isotonic contraction. Isometric contraction is this. So we have again our humerus, and we have again the angle. When let's pretend that this weight here is too heavy. Let's pretend that, I don't know, 100 pounds, something like that, and you're not able to lift that weight. Even though you're trying, your muscle is still contracting, but because the load is so heavy, there's not net movement. So there's still 90 degree. So overall, the load didn't move, and that is an example of isometric contractions. So when you try to lift something that's very, very heavy and barely can move, you're performing isometric contractions. Now, other examples in which we have isometric contraction is muscles that in your back, for example, that will keep your posture. Those muscles will contract, but it will not make enough movement, just enough to keep your posture. And then the isotonic contraction, of course, is the ones that we use most of the time. And there is there's two types of isotonic contractions. The one there is called concentric contraction and eccentric contractions. Remember, in isotonic, there's a change in the angle. So in concentric contractions, the angle is reduced, like in this example. In eccentric contractions, the angle is actually getting bigger. So let's pretend that we start here. We have a 90-degree angle, and then we still, if we follow the example of lifting weight, we'll still have some contractions, but in opposite sides. So let's pretend here you have 110 angle. So you increase the angle in the eccentric. In the concentric, the angle is reduced, but overall in both, they're muscle contractions. One, the muscle contraction is pulling, and this one is pulling towards the origin of the muscle, and in this one there's a still contraction that is the contraction is getting less powerful, that even though your muscle is still contract, the movement is in the opposite direction. Now let's talk about the motor units. These are very important because I told you before, muscles cannot contract without a neuron that stimulates them. And the motor units are very important because they're the parts of the neurons that attach to different muscle fibers in order to contract them when is needed. So to explain the motor units, I bring this picture here. The whole thing here is a muscle fascicle, and each of these are muscle fibers. Remember, muscle fibers are essentially the muscle cell. So we have two types of motor units, and I'm going to say this: the motor unit again is a group of muscle fibers in a muscle innervated by a single motor unit. So it's a group of muscle fibers that has been innervated just by one single motor neuron. We have two types of motor units. We have a small and we have large. So in this part, I'm going to make a small motor unit, and in this slide, I'm going to make a large motor unit. Now, I told you that from the spinal cord, we have the neurons, or we have a nerve, right? So for example, we have a nerve. Let's think about a nerve, like medial nerve or radial nerve or the ulnar nerve. And those nerves are not just one, but there is many neurons inside of a nerve. So in pretend all these lines are neurons that connects and innervate the muscle. So I'm just going to take one of them. So I'm just going to take one as an example. Remember, we have more. I'm just going to draw a few more, but I'm just going to get one just so you know. And this neuron will get here, and then it's going to spread some of those dendrites and are going to do those neuromuscular joints. So for example, this is just one neuron, because innervates 1, 2, 3, 4 muscle fibers. That's a small motor unit. So it's a single motor neuron that supplies a small numbers of muscle fibers. And those small motor units are very important for fine motor control. So fine motor control is when you do things that you don't require a lot of strength, but requires precisely or precision, or there are movements in which, for example, when you type in your computer or in your phone, you're not using the whole muscle, you use just a few muscle fibers from the small motor unit. Then we have the large motor unit. Again, let's pretend, and the large and the small motor unit can come from the same nerve. So let's pretend that this is a large one. So what's going to happen here is that this kind, it will attach or will send projections to many more muscle fibers. So if you can imagine, when this neuron here, the large motor unit, gets stimulated by an action potential, the action potential will travel and will stimulate all of those muscle fibers. So when those all muscle fibers contracts, therefore the movement is going to be more gross or more strong movement. So large motor units are associated with gross movement. And like again, it's a single motor unit that supplies a large number of muscle fibers. So in your hands or in other muscle, we have a combination of both, and the body will determine when we're going to use the small ones. For example, in your hand, for example, if you want to do something that is precise, not strained, but like typing or holding a mug or holding a glass of wine, whatever, you might use a small motor units. And then if you want to, squeeze an orange, do something that requires more powerful, you might use large motor unit. And in some circumstances, you're going to use both of them. Now, other topic here that is important about how your nervous system influences in your muscle contraction is the frequency of the motor neuron stimulation. So we have three types that are important. Well, let's get started here with the twitch. The twitch is a single contraction of a muscle fiber triggered by a single action potential from a motor neuron. So when one neuron sends an action potential and there's just one contraction, what is going to happen in terms of, if we want to put this in a graphic, what's going to happen is this. So this part here, and if this is time, and if this is tension, what is going to happen is the action potential from the neuron start here, and in the muscle is going to be a period of time where all these channels are going to start opening and muscle contraction will begin. Then when ATP and calcium and those myosin and actin react each other, there's going to be the part of the contraction that is actually stronger. And then if the action potential is terminated, or if you have just one action potential, that calcium, that acetylcholine that is released in the neuron will be absorbed by the acetylcholinesterase, and the stimulus will end. So therefore the calcium will be removed from the cytoplasm, and the troponin, tropomyosin will block the binding sites, and then relaxation will occur. Now, when we have multiple stimulus, we're gonna have something like this, like a wave summation. It's called summation because it's a zoom or twitches. So the rate at which a motor neuron fires action potential affect the tension produced in the skeletal muscle. So let's see that you have these. I'm going to explain this here in this graphic. So we have one twitch, but let's pretend that when this relaxation is going to start, other action potential is triggered. So other muscular fibers will be stimulated, and then we're going to have this period of elevation, contraction, and then when it start relaxing, other action potential will trigger another one. So if you can see at the end, the zoom is that the tension will progressively get higher through time. So that's what we can see here. So if you, for example, you're trying to squeeze an orange, right? So you start with a little bit of tension, And then when you want to push and squeeze more, this tension gradually will go up until, you know, two things happen. One, you decide stop that contraction, or until your muscles gets fatigued, and then you have the relaxation all the way to the starting point. That is wave summation. Then tetanus, this is something that happens normally. This is abnormal, and if you see in twitch and wave summation, we have those small periods of relaxation. What happened in tetanus is that when the stimulus frequency is so high that relaxation phases disappear completely. So in tetanus, we're going to see this. The stimulus is going to start, and then it's so strong and continuous that the muscle will keep contracting with no period of relaxation until the muscles essentially burn out or fatigue completely, and then they're going to relax. This type of tetanus frequency happens when you have a disease. It's called tetanus disease, and we have the characteristic involuntary contraction of the muscles that sometimes can last several minutes. You cannot control it. Your muscles get contracted so hard that in some point even you can have damage of your muscles for those tetanus contractions. The muscle tone, that is important, because when we are not thinking about contracting our muscles, about moving the arm, the muscles always have a tone, and the tone is what make the muscles, or keep the muscle shape, if you will. So muscle tone is important to keep your muscle shape, and is if we don't have muscle tone, our muscles will be flat and completely relaxed. So really, muscles never are completely relaxed. There is in a point of some of the muscle fibers might be having some sort of contraction, and then there's other muscle fiber will interchange between contractions. So that will keep overall your muscle tone. Muscle tones, as you can imagine, will not fatigue your muscles, because you have time between when one group of muscle fibers will contract a little bit, others will be resting. When those that has been contracting will rest, another new group will contract, and overall that's going to make your muscle tone. When you have absence of the muscle tone, that is hypotonia. And hypotonia, we have a muscle that is flaccid and very weak. There are some abnormal diseases that can cause hypotonia, and when you have that, also you have weak reflexes. The opposite is true. When you have a tone that is very high, it's called hypertonia, and you're gonna have some hyperreflexia. Now let's talk about the type of muscle fibers, exercise, and muscle performance. We have three types of muscle fibers according to the metabolism that they use to generate or produce ATP for muscle contractions. We have the slow oxidative fibers, we have the fast oxidative fibers, and then we have the fast glycolytic fibers. Now in the slow, as the name implies, the fibers contract relatively slowly, and therefore, because they contract slowly, the metabolism they're going to use is aerobic respiration, so oxygen and glucose to produce ATP. The fast oxidative fibers, of course, they have fast contractions, and primary they use aerobic respiration, but they may switch to anaerobic respiration. Therefore, when you have anaerobic respiration, remember we have lactic acid building up, and that might cause more muscular fatigue than the slow oxidative fibers. And of course, I told you that when we breathe, we get oxygen. oxygen is what we need for aerobic respiration. So because those slow oxidative fibers are moving relatively slow, the oxygen that you're breathing is the demanding for oxygen is not that high, so you will have in theory pretty good amount of oxygen. And the fast one, you might have oxygen, but then in some point your muscle keeps contracting harder, therefore they need more ATP, and the oxygen that you're receiving through your bloodstream when you breathe might be not enough. So the rest, the rest of the metabolism is going to turn to the anaerobic, that metabolism that will not use oxygen, and that will lead to muscle fatigue and lactic acid building up in your muscles. The other type of fiber, muscle fibers, are the fast glycolytic muscle fibers. Those has fast contractions, and primarily they use anaerobic glycolysis. Now let's talk about exercise and muscle performance. Now, although muscle cells can change in size, new cells cannot be formed when muscle growth. So skeletal or cardiac muscles are not able to regenerate. They're not able to make new muscle cells. Now, the way how the muscles grows in those, as you can see, for example, in that picture, this guy, this is called hypertrophy. This is when the cells grows in diameter or increase in diameter, and the cells are able to increase in diameter because an increase in the structural proteins and the muscle fibers. So somebody who do exercise, those muscle structures are going to be proteins like myosin and actin. There is going to be an increase in the sarcomeres, and all the proteins or structural proteins inside of the muscle fiber will increase. So therefore the muscle fiber, instead of being this side, it might be I don't know, this side. So that is called hypertrophy. Now the opposite of that is called atrophy. That's a decrease of the muscle mass due to loss of structural proteins. And when we are old, this is called age-related muscle atrophy or sarcopenia. Remember, sarcopenia, it comes from those sarcomeres that we have been losing. So in atrophy, you see here a picture how a normal muscle will look like, and then a muscle that is atrophied looks like this, like with less muscular mass. Remember, in age and old people, they have sarcopenia, or sarcopenia is essentially a type of atrophy. When we talk about exercise and muscle performance, it's important to know that we have two types of exercise that will determine the performance of the muscle contractions: the endurance exercise and the resistance exercise that we can do. The important parts here is that endurance exercise, most of the time, are slow fibers are predominantly used for endurance exercise. Now, because also in endurance exercise, the type of exercise that we're going to do is progressive, that give us time, or give the muscle time to use more oxygen and aerobic respiration. Also, in people who do endurance exercise like running, aerobics, swimming, or going the bike ride or stuff like that, those people will increase the amount of myoglobin. This myoglobin is a muscular protein that the function is that it holds or storage oxygen. So it works as a reserve of oxygen. And if you remember, when we don't have oxygen, when we don't have enough oxygen, we don't perform aerobic respiration. Instead, we do anaerobic respiration. And remember, anaerobic respiration and lactic acid building up in your muscles, and that cause fatigue, muscle fatigue. Other thing that in endurance exercise we have angiogenesis, which is the creation of new blood vessels. So that overall is a thing that requires time. But if you can imagine, if you have a muscle that is constantly working, constantly contracting, like the muscle here in the legs of those guys, those muscles and those people have a lot of blood supply. And if they have more blood supply, that means that more oxygen can be oxygen, and also waste products can be interchanged, can be transported uh, through those blood vessels. So anyway, endurance exercise, the characteristic is that people who do endurance exercise have more myoglobin and angiogenesis. And resistance exercise, they require large amounts of remember those fast glycolytic fibers to produce short but powerful movement. So for example, somebody who is lifting weight in the gym to build up muscle, the type of exercise he's doing is resistance exercise. And most of the time resistance exercise is usually, most of the time, there's not significant increase in mitochondria or myoglobin, or increase in capillary density or angiogenesis. Now let's talk about cardiac and the smooth muscle. I told you that we're going to talk about these in more detail in AP2 and AP2 when we see cardiovascular system. And then the smooth muscle we're going to talk about in more detail when we see the different systems like that, digestive or reproductive, urinary, etc. So here we're going to just mention the most basic and important information for your exams. Let's talk about the cardiac muscle tissue. I told you that we can only find cardiac tissue in the heart. What makes the difference between the skeletal and the cardiac? Both of the cells make striations. Both cell has sarcomeres. Both cell has sarcolemma. Pretty much everything, except that these ones, the cardiac cell has just one nucleus, and this is the important part here. The cardiac cells contain those intercalated discs, which is essentially unions or junctions between cells. So if you take a look here, this is a single cardiac cell, and this is another cardiac cell. So between those two, we have those intercalated discs. If we take a look of those intercalated discs closely, we can see that we have desmosomes and we have gap junctions. So when one cell, let's pretend that this is one cell here, and this is another cell here. So this is cell number 2, this is cell number 1. When the action potential travels through here, it's going to go here. It won't be able to stimulate the cell yet, but remember, gap junctions are called gap junctions because they have like a gap, or they actually allows nutrients or things pass through. So through those gap junctions, this second cell will get the action potential, and the action potential will be able to spread to another one. So when you stimulate just one or just a few of the cardiac cells, you are going to be able to stimulate the whole cardiac muscles, and that's how the heart works as a pump. Because with one stimulus, you'll be able to stimulate many of the cells. Remember that in the skeletal muscles, we have muscle fibers, but you need neurons that innervate each of those in order to contract the 3. Here in the heart, you just need to stimulate one, and because you have those intercalated discs, that one stimulus might be able to stimulate the rest. The smooth muscle is a little bit more complex. Like I said, we're going to just mention the general things that you need to know. First, it's called smooth because do not have striations. So that means that they don't have those myofibrils in the way that normally skeletal and cardiac muscle has. We still have, of course, actin and myosin. It's just that the way how those proteins are arrangements is different. Other thing is that smooth muscles is present in the wall of the hollow organs, so all the organs like the stomach, even in some part of your eyes, the ones that make your pupil be small or big in the darkness, that is a smooth muscle, and also in your arteries and veins. And then the single smooth muscle cell is like a spindle shaped, so it's wider in the middle, and then in the borders is a little bit more narrow. So if I have to draw it, it's something like that. So it's wider here and then goes a little bit narrow in this area. Other important thing is that here I told you that the smooth muscle contains actin and myosin. Those are crucial to contract. So a smooth muscle, therefore, it has to have those in order to contract. But they, instead of having the sarcomere, this smooth muscle will have the equivalent of that. It is called the dense bodies. Dense bodies are all these here. So that is why the smooth muscle has no those striation. If this will be, for example, a skeletal muscle, all you will see something like this, and those will be the sarcomeres. Those will be the things that make those striations. So here in the muscle, and the smooth muscle, we have those dense bodies. Other important thing or difference is that smooth muscle do not contain troponin. And remember, troponin and tropomyosin are the ones that block the actin binding site. Instead, what happened is here, the protein that is in charge to the regulation of the contraction is called calmodulin. And this calmodulin is the one that calcium will attach. So when calcium attaches to calmodulin, it will activate the head of the myosins. So again, what is going to happen is that instead of having, the head of the, I'm going to draw here, myosin, and we have the head of the myosin here. In the skeletal muscle, remember we have ATP that it keeps this more like in this direction. We have the same thing here, but in a skeletal muscle, the only thing that you need in order to make this head of the myosin attached to the part of the actin is just, that you don't have those binding signs blockaded. So here in the smooth muscle, because we have the calmodulin and we have the calcium, the head of the myosin and the smooth muscle is inactive. So you need, in order to activate this head, you need some phosphate group. You need a phosphate in order to activate it. And the enzyme that is going to attach this phosphate to the head of the myosin is the calmodulin. And specifically, when calcium attached to the calmodulin and that complex is formed, that will activate this enzyme that I mentioned, and that enzyme will add the phosphate to the myosin head, and then the myosin head, it will be activated and therefore will be able to attach to actin, and therefore contraction will occur. I told you that smooth muscle and cardiac muscle is not under voluntary control. A smooth cardiac muscle and skeletal muscle, but mainly skeletal muscle, usually can be triggered only by neurons. Now, in the smooth muscle, there are things different that can trigger contraction of the smooth muscles, like hormones, for example. In childbirth, oxytocin is a hormone, and that oxytocin stimulates the muscle in the uterus to contract. So hormones can stimulate the smooth muscle. neural stimulation, so nerves, and specifically the part, because it's involuntary muscle, the autonomous nervous system will activate the smooth muscles, and other local factors like distension, for example. If you think about your intestine and you have food that is passing through the intestine, that food, when it's accumulating there and is making pressure in the walls get distended, and that triggers muscle contraction, so the food or whatever can move and pass. Other thing that is important is that this is the only muscle tissue that can create new cells. That process is called hyperplasia. This is the only smooth muscle that can create more smooth muscle as needed. And you think about this also during pregnancy or during childbirth, when the uterus is growing, growing not just in size, so the cells are not growing just in size, but also in amount. Finally, let's talk about the development and regeneration in the muscle tissue. The important parts here that you need to remember is that the whole muscular system is coming from your mesoderm. Remember, we have ectoderm, endoderm, and the mesoderm will make all your muscle tissue. The the stem cell that will make your muscle during development is called the myoblast. And if you remember, during development in the skeletal muscle, many of the myoblasts cluster and fuse each other, and that's why you have a big cell, big muscle fiber with multiple nucleus, because those nucleus belong to an individual's myoblast that has been fused. Although the number of muscle cells is set up during development, we have satellite cells. Those are the cells that are related with repairing skeletal muscles. The satellite cells can, in a very limited extent, can regenerate some of the muscle fibers, but the satellite cells just help to repair damage. Cells that are still alive. Most of the time, if a cell is died, is virtually impossible to create a new muscle, skeletal, or cardiac cell. So instead of that, the satellite cell will be able to repair some of them, and when it's not possible, what is going to happen is the cell is going to die. The muscle fiber is going to be replaced by fibrous tissue. So fibroblasts are going to create fibrous tissue, which is a type of connective tissue. In your heart, for example, when you have a heart attack, the cardiac cells are die because of lack of blood supply, and then they're going to be replaced with fibroblasts, which of course will not be able to contract. So somebody who has a heart attack and infarct, the part of the muscle that is dead will never be able to contract again. And then I told you the only muscle can regenerate is the smooth muscle and the stem cell, and the smooth muscle is called the pericyte. I hope this video helped you to understand the physiology of the skeletal muscle better. Thank you for watching, and I wish you a very nice day.